Well, it's a pleasure to start these festivities off this morning by telling you a little bit about my experiences with Susumu Tonegawa over the years. I first ran into him at a symposium in Germany. It was one that was held by the Biochemistry Society there every 2 years in Mosbach. And it was my job in 1976 to summarize the events of the meeting. The big event of that meeting was a then young, unknown speaker named Tonegawa, who was here, who was there at the meeting from Basel, Switzerland, where he was at the Basel Institute, where he described an experiment that really changed the course of immunology from then on. The details of that work, Lisa Steiner is going to summarize when she follows me, because I'm going to just talk about some more personal anecdotes. I had to summarize that meeting, and a lot of interesting things going on. I was always very proud of the summary because it caught the essence of what Susumu had done and to recognize the importance of it at that time. A few years later, I was on a committee at Harvard that was looking for an immunologist to establish a big presence there in Cambridge. And I and another older immunologist were the two outside people on the committee. And we both, of course, suggested Susumu. And they all went for that. And for a time, it looked like he was going to go to Harvard. In fact, I think the Harvard Crimson published it. But some details went on during the times of the conclusion of that meeting, and there's some cloudiness about whether he accepted or didn't accept. Anyway, luckily he ended up here, not down the river. And so there we had the opportunity to interact with him, beginning in about 1980 when he came to MIT. But around that time, there was a tremendous amount of interest in the immunological community in identifying the receptor on T cells that was antigen sensitive. A big event happened with, one of the things that happened was that Tokmak stumbled on a gene for what turned out to be the beta subunit of that receptor. Mark Davis, through a very systematic and rational approach, found it also. But the big problem was to find the other subunit because the complete receptor was known to be a heterodimeric molecule, two subunits, called already alpha and beta. Susumu had a Howard Hughes investigator at the time. And I remember meeting at Howard Hughes that I was on their advisory board then. And the big push was to find something like the heavy chain, the immunoglobulin heavy chain that might be the beta subunit. And Susumu had a number of interesting experiments going on. But we wondered about the cells he was using as a source of the cDNAs he was making, I guess. And we had we were cloning T cells around that time, and one of them was really growing very well, a clone called 2C. And so those cells were then used by Susumu. And Dave Krantz, who was a postdoc in my lab, did something pretty heroic in terms of expanding the growing of that T cell clone in large amounts. And with it, Susumu, and particularly some people in his lab, Hiro Saito, and Dave Krantz, who said he'll grow all the cells and do all of that. If Susumu will teach him, we'll let him learn how to do the cloning, which Dave joined in with. And out of that came a paper with the two subunits, genes for the two subunits, and a paper was published in Nature very rapidly, a very triumphal note. In fact, I remember the paper was written in a great hurry because this was really an intensely competitive field at the time. It was written in a great hurry. I remember on a weekend writing the second half of it. Susumu did all the first half and everything else, and it went together seamlessly, went into Nature, was out, I don't know, 2 weeks, something like that, 3 weeks. And Susumu was a big demand on the lecture circuit. and went out talking about it. Dave Krantz in my lab did a two-dimensional, 2D gel of the T-cell receptor using an antibody that must have been available at the time. Lo and behold, in this 2D gel, both subunits had a typical ladder of glycoproteins, like N-glycans. But when we looked at the sequence of the alpha subunit, which is what Dave was focused on, there was no triplet that could be glycosylated in the right way. So I remember Susumu was away talking about this. I called a carbohydrate chemist I knew very well in St. Louis, Stu Kornfeld, and asked him whether you can get that kind of N-glycosylation look, that appearance in a gel, for a protein polypeptide that couldn't be glycosylated on asparagines. Stu didn't think so. So it was pretty clear there was some kind of problem going on. And when Susumu came back and we broke the news to him, of course he was a bit disturbed at the time, as you can imagine, to put it mildly. But to his credit, what he did was immediately go back to the library himself, not through Site or anybody else, and fish out the alpha subunit, or the clone for the alpha subunit. And at this point, the paper got written very quickly, and there was the two genes for the complete T-cell receptor. The previous paper also called the T-cell receptor and the complete primary sequence of it, deduced from the cDNAs. Turned out to have another gene called a gamma gene for another T-cell receptor, which then is a whole other long story. But it was that behavior that was pretty interesting, so reflecting a lot about the tempo of the time, the competitiveness, how one reacts under pressure, what one does with an error, just plunges ahead and tries to correct it. And I thought that was pretty interesting set of events. Let me stop now and turn this over to Lisa, because she's going to talk about some of the scientific basis for Susumu's important announcement in 1976.
Okay, second part of this team. Like Herman, I also met Susumu for the first time not at a meeting, but at a seminar. He was invited at around that time by Alvin Pappenheimer at Harvard to talk about his work. And I heard about the seminar, and I guess I'd heard about this exciting work, and I rushed over to Harvard and heard the seminar, and it was extremely exciting. And I remember we went, Pappenheimer, Pap, and I and Susumu went over to have a drink at the faculty club post-seminar. And it was a great occasion for me because I was really so excited. Why was I so excited? So I'm going to spend just a few minutes explaining that. I guess it was primarily the work of Karl Landsteiner in the 30s and 40s that demonstrated the incredible specificity and diversity of the antibody response. And the central problem for immunologists after that was to explain this on a protein and genetic basis. This was very difficult for many reasons, but one of them on the protein level was that antibody responses were always heterogeneous. So you couldn't actually get your hands on a homogeneous protein to study. This was before monoclonal antibodies came on the scene. Rodney Porter in the 50s showed that you could break up the immunoglobulin or antibody molecule into two pieces, one of which, FAB, bound to the antigen, the other, FC, crystallized, which is why it was called FC. Now that's very peculiar because here you have a heterogeneous antibody. It was a rabbit antibody, and it certainly won't crystallize. It's a mixture of molecules. You whack out a piece of it, and that crystallizes. That was very astonishing. So that was kind of the first indication that the antibodies might be composed of two parts, one part that varied and bound antigen, and one part that was constant. Next major step was Porter and Edelman described the 4-chain model, there were 2 heavy chains and 2 light chains in the antibody molecule. And it was soon shown that indeed each chain had a part that varied, the variable region, and a part that was constant, the C region. These were the first hints that there might be something peculiar about the genetics of the antibody molecule, because how could you have a protein polypeptide chain, one part of which varies all over the place and the other part is absolutely constant? There was no idea of a gene that would do this. And so immunologists, and I think nobody else paid much attention to them, soon began to say that there must be 2 genes specifying the antibody molecule. The V and the C regions are under separate genetic control. But how this might work was anybody's guess, really. One had no idea. So the next step then was there was a great deal of argument about these presumed genes that were specifying the V region. Were there thousands, millions even, of variable genes that were inherited that all of us pass on by heredity in the ordinary way, which seemed like kind of strange? Or was there just one or a few genes that varied somatically during the lifetime of each individual? And people debated this very heatedly during the 1970s, I guess that was. So this was kind of the general background to the work. And Susumu then, as you'll hear, applying methods of molecular biology, was able to demonstrate that there were indeed separate genes for the C region and the V region, and that during the development of the B lymphocyte, these genes are rearranged to make the antibody, the gene that then encodes the protein for the antibody molecule, both the heavy and the light chain. So this work really explained so many things. It was just I can't tell you really how exciting it was for people in the field and for people elsewhere in biology. On a minor personal note, my lab had struggled for years to determine the sequence of a heavy chain of a crystallizable myeloma protein. And by a lot of hard work, we managed to show that this protein was missing the hinge region right in the middle of the heavy chain. By classical protein sequence, it's very hard to find such a thing. I mean, it sounds easy now, but it was very hard then. And we couldn't understand this. We finally, after years of work, had the data, and we just were unable to understand how could this particular hinge region be missing. Well, it was also Susumu's work showing that the heavy chain constant region had different different what we now call exons, specifying each part of the constant region, including the hinge region. So that could easily explain either a deletion or a splicing error of some sort, why we had we were missing. If you compared it to his published sequence, it was exactly that hinge exon that was missing in our protein. So we went ahead and wrote up the paper after years of sitting on it. Without further ado, I'll give Susumu his turn.
Okay, so I find this program is great. Okay, I mean, we'll see how it goes, but I mean, it's a joke, but I think the part biology is really getting humanized and culturalized, and this is the part of that direction, and I'm very pleased to be here. So I don't exactly know what I'm supposed to do because I have never had the chance to attend the previous ones, Jellyfink and so on. So I'm guessing. So we have about 45 minutes, and my understanding is it'd be good if we save some time for question and answers because it's supposed to be conversations, right? So in any case, I give you a little bit of a sort of a lead for conversations. First of all, many years before most of you are born, except Herman Eisen, I was a student at University of Kyoto, Kyoto in Japan, and I was in chemistry major. But toward the end of the 4-year schooling, I wasn't sure whether I want to stay in the field of chemistry. And of course, chemistry turned out to be it's still very exciting field, but I did not know enough of it. And I was looking for something new to do for graduate school. And someone told me that this is 1962, so many, many years ago. Someone told me that the new biology emerging in America, primarily in the United States and partly in Europe. And that is called molecular biology, okay? And I had no idea what that is, but he introduced me to a couple of papers he recommended me to read. And those papers were by Jacob and Monod from Pasteur Institute. And I hope biology student knows these names, And so I read those papers in a journal called Journal of Molecular Biology, which was a Cell of today at that time. Okay? And the long paper, it's operon theory. And I was so impressed by this, and I immediately, instantaneously decided I'm going to be trained as a molecular biologist. Okay? So, but in Japan, there was no molecular biology lab at all. Okay? So my professor was kind enough to arrange me to study abroad, and I ended up one of the most beautiful towns in the United States, which is La Jolla. So UCSD was just starting. There's only one more year above me in a graduate school at UCSD. I'm the second year in the history of UCSD. There was no undergraduate student at that time, so it started at the graduate school university. So I was trained as a molecular geneticist, molecular biology, molecular genetics. And at that time, unlike now, major thrust in molecular biology was not in eukaryotic systems, but in a prokaryotic system, bacteria and viruses which infect bacteria, phages. Okay? So I worked on phage lambda, transcriptional control with some genetic components into it at my PhD thesis. Thesis was okay, but not something I'm particularly proud of. But then in the late 1968, so I was going to go to postdoc. Now, at that time, among students, my fellow students, there was some concern about the future of molecular biology, okay? Because it looked like the major fundamental problem in molecular biology, molecular genetics, are being solved. Okay? Genetic code, protein synthesis, messenger RNA, all of those things are discovered. And we were wondering, is there a future for us when we become independent after several years of postdoc training? So among us, my close surrounding, that we should go to a more complex system. Okay? So the animal cells or plants, something like that, to study a development, differentiation, cell growth control, and things like that. But at that time, there was no really good technology to do that in order to do rigorous research as it was done in prokaryotic system. Okay? So it was a difficult decision to make as to where to go for the postdoc. And I heard of this institute called the Salk Institute, which is right across the street from North Torrey Pine Road from UCSD. And there was a lab, which is run by someone called Renato Dalbecco. Now, amazing thing is, among young people here, I don't know how many percent of you know who Dalbecco is. Those who know, please raise hands. You see, it's only older people. We don't use the Dalbecco medium anymore for tissue culture. We still use that. So people know by the medium named after him, but that's not his major contribution to to research science. And he is a biologist, actually. But he comes from this old school of molecular genetics, molecular biology, which is sort of started in Caltech and a few other places. Max Delbruck was a leader, and Beck was already there, the young faculty member. So he's the MD, he's a biologist, but he was already exposed to what used to be called molecular biology. So he had a very lab, and his main interest was tumor biology, to study role of viruses in tumorigenesis. So there, you use the tissue culture of animal cells, okay? And then you have a virus, and they were, at that time, focusing on small genome virus, simian 40 virus, polyoma virus, okay? So there are no more than 4 or 5 genes in there. You can purify DNA, viral genome, and then you can infect in a culture, and a condition where to go the lytic cycle, where the cell or host will die, or you can, on a different cell type of cell, you can transform them to make it out of control for growth. Okay? So it sounded to me, it is a sort of hybrid between the molecular genetics of prokaryotic system, which we are used to, and at the same time, you can learn something about the animal cell, the regulation in animal cells. So I chose to apply that place, and fortunately, Renato took me, and afterward, I found out several of the postdocs who joined the lab around the time I joined, they had exactly the same consideration. They worried about their future, and they wanted to learn, work on the eukaryotic system, but not the purely, animal system or plants or something like that. Okay? So that's how I was exposed to the eukaryotic system, eukaryotic system. And I also learned a lot in that lab, but although I was there only 20 months. And I tell you why I was there only 20 months in a minute, but lab was fantastic. There were very good postdocs, senior postdocs, the big lab, like 30 people. And Renato doesn't, when you go there, Renato doesn't tell you what to do, okay? He will say, oh, why don't you just talk people and try to find something interest you. And then he said he'd happy to discuss it if I bring some project ideas, but he wouldn't tell you what to do. And I learned a lot from these postdoctoral people, very good people like Joe Sandbrook, Heiner Westphal, and those people who later became a very good senior scientists. Anyway, so I learned something there, but it was relatively short, as I said, like less than 20 months. And the reason why it was less than 20 months is because I came to UCSD with a visa called J1 visa, exchange visitor visa. And nowadays, there is some way to get around a little bit, but at that time, it was very strict. So after 18 months after you finish PhD, which is called, this period is called practical training, you have to leave the country, okay? You have to go, you don't have to go back to your native country, but you have to go out of the United States for at least 2 years, to come back. So I had to leave the US. I was very happy with what was going on in Renato's lab, but I had to leave. So Renato was concerned, very nice to me, and he talked to some people in Canada, because Canada is close to the US, and then if I stay, at the shelter there for a few years, I may be able to come back. No, you may laugh at this, but there are a lot of people did that at that time, okay? So, but my postdoc work was not so spectacular, so I could not expect to have, best faculty positions. But I ended up having offer assistant professorship in a small town about 2 or 3 hours east of Montreal, and the town is called Sherbrooke. Okay? It's a small town with a large hospital, and there was some research division there. So I got an offer. I wrote a grant, MRC grant, Canadian MRC grant, and I got it. And I was supposed to go in about three months. When I received a letter, short letter from Renato, who was traveling. He's Italian American, so he often goes to Europe. He was traveling in Europe, and I still have that letter. I could have brought a copy, but I didn't do it. It's written in a small stationery with a logo of a very famous hotel in Rome. It's called Hotel Hasra. Hotel Hasra is just above Spanish Steps. Okay, it's still there. So he wrote this handwritten letter to me, only 1.5 pages, small ones. And he said something like, I don't know what you decided about the place you have to go after you leave my lab. But if you have not completely decided, I want to suggest an alternative. That's what he said. And he said something like, he believes, this is 1968, okay? 1969. He said he believes time is ripe for young scientists with good molecular biology training to go into immunology. That's what he said. And if you are interested, he says, write to Dr. Director Niels K. Jane, 487 Grenderstrasse, CH 4058, Basel, Switzerland. Okay? And when I saw that email, the letter, I took it downstairs in the Salk Institute where Mel Kohn, you know Mel Kohn. Mel Kohn is supposed to be also immunologist, but also come from molecular biology. I went there and showed this letter to a postdoc whose name is Jim Watson, okay? But not the one you know, but another one. And because I knew him, and he comes from molecular biology, so I had some affinity to him. So I asked him, is there anything I can do in immunology which could be significant? And they said he had some molecular biology background. And he immediately said, no, there's nothing. Okay? So I went back and put it in my letter in my drawer, and I forgot it. And then about a month later, and it was almost 2 months before I was supposed to be deported unless I voluntarily leave, Renato came back, and he came to tell me, he asked me whether I wrote a letter to Jane. So I explained to him that I did not, and I told him I don't know anything about immunology, and I don't even know where Basel is. I didn't know who Niels Jane is. So I didn't think this is a good idea, I told him. But now, I want to tell you something. It is very important. Renato, as I told you, doesn't tell you what to do, usually. And, they even do not recommend strongly to do anything, okay? But this case, some way, and it struck me, because he said to me, Susumu, this is really interesting direction. You should really think about it, or something like that. So, then about 10 days later, I visited Montreal, which was end of December. And I was supposed to start in April, but I visited, and it was damn cold. So I came back, and I just, my mind flipped. I said, okay, I'm gonna I'm going to trust Renato, and I'm going to go to Basel. Okay? So actually in February, I left and went to Basel. And my intention was, again, just stay there for 2 years and continue to work on. I was making some polyoma virus mutant, temperature-sensitive mutants, which would be defective in transformation, cellular transformation. So I wanted to continue that research. Fortunately, Janne allowed me to do that, although when he saw me, a week after I arrived, he asked me whether this had anything to do with immunology. So I said, no, I can't think of any connection. And the second thing he asked me is, do I know the difference between T cell and the B cells? And I had never heard of that. So I said no. And, but he did not fire me, and he let me do. And in Basel Institute, actually they had this, what is called horizontal structure, which means if you have a PhD or a PhD equivalent, you're independent, okay? It doesn't matter whether you haven't had any postdoc period or not. And you can collaborate with anybody, but you are given one technician and half of a room, half of a lab, and half of a office. And you can do whatever you want to. And you're supposed to do something relevant for immunology. But I was ignoring that. But in about 2 years, I suffered, actually, because I didn't understand what they are talking about. There were a lot of seminars. Apparently, there were very good immunologists to assemble there, but there was no molecular biologist or biochemist or anything like that. It's pure immunology. At that time, dominant approach of immunology, what is called cellular immunology. Okay? So there are seminars, and I mean, I tried to go, but I didn't really understand. And so, but gradually, my thinking sort of changed. I thought, if I'm going to stay here, surrounded by apparently very good immunologists, I should learn something in immunology, and I should try to do some research which interests them. Okay? Otherwise, I don't have opportunity to talk with them, science. So I fished around, and I ran into this problem of genetic origin of antibody diversity. When I was exposed to this problem, I actually, honestly speaking, I didn't understand why they don't study antibody genes rather than antibody molecules themselves. Because the only thing people were doing at that time, as Lisa said, but a lot of other people like Lee Hood and other people, Melcon, they were sequencing myeloma proteins, which is the monoclonal immunoglobulin proteins. And they are hoping that if you sequence enough of them from that pattern, you may be able to answer the genetic origin of antibody diversity. And I thought this is crazy. I mean, how can you do that when you have to look at the gene and compare the genes with the proteins? And just on one side, it's not going to solve the problem. And fortunately, when I was in Basel, Renato's lab, I already knew about the restriction enzyme, and also almost gene cloning is almost coming. That was the great thing about Renato's lab. Renato's lab in tumor biology was the mecca in the tumor biology field. So a lot of people actually sent preprints to Renato's lab to, I guess, to get some opinions or something like that. So I already knew this, I think 1968, first paper with Smith and what's the other guy? I forgot. Restriction enzyme applied to Dan Nathan, Smith and Dyson. So I thought, when I heard that, I immediately thought the idea that I executed later, that if you cut up genomic genes, antibody-producing gene versus non-antibody producing genes, if there is any DNA rearrangement, if we cut them up, and if we have a probe, by the way, hybridization probe, run the gel and the probe pattern, basically Southern blot, but Southern blot didn't exist at that time, okay? So that was a technical issue of how to do Southern blot equivalent, because it didn't exist. But theoretically, if you can do something like that, one should be able to tell whether gene is rearranging or not automatically. Okay? So I started getting myeloma cell line to grow them in the song, but at the same time, I was always thinking about how to spread out, restricted the genomic DNA. And we did something really non-elegant, okay? Because Southern blot is, as you know, it's very elegant. But we ran people who works on the serum, they separate the proteins in the serum. They use electrophoresis, horizontal agar electrophoresis. And if we wanted to do preparative things, they use very large one, like 50 centimeter long and maybe 40 centimeter wide or something like that. You have to run a long time because it's a very large gel. And I saw that in the cold room, the immunologists were using. So I thought, oh, maybe I can use this one, put the DNA, the restricted DNA, and develop it electrophoretically. And there were a lot of trial and errors, but it turned out you have to do it very slowly because otherwise, if you rush, agarose get constricted and distorted. It takes about 3.5 days to run. But, and then you cut it up. And then extract DNA from each fraction, like 60 fractions. It's not the Southern blot. And then they do the hybridization with a probe, immunoglobulin gene probe, which itself, there was no cDNA then. So what we did is we purify mRNA as much as possible from myeloma cell line, the specific immunoglobulin light chain mRNA. I don't think it went any more than 97, 98% pure, even if we did many steps of purification. But then you iodinate, okay? So to label, you iodinate. And then use that as the probe. And, so that's how it went, okay? So I don't tell you all the detail, but we have only 20 minutes left, so maybe I should stop here. And the only message I want to give to young people is I'm so grateful to Dalberto, okay? And I think he is a most sophisticated mentor without any intentional planning. He had this, in my view, he had this vision, okay, where the science is going, where the exciting things are going to develop in 5 years, 10 years, maybe 30 years. And he was not an immunologist, but somewhere he knew enough to suggest to, I guess, ambitious young student, postdoc, to try. Okay? And I think, as a matter of fact, I'll tell you a little bit more, because he actually, in the letter, I abbreviated, but he suggested something very specific. It's called immune RNA. Okay? Now, there was a claim was made around that, just before that, one group in Harvard and one group in NYU or something, independent. If you take a rabbit, immunize with antigen, and then afterwards extract RNA from spleen, and they inject that RNA to naive rabbit, that rabbit become immune to that antigen. And that RNA is called immune RNA. Okay? But it's very crude. I mean, you just take RNA and inject it. And there were two papers. And Renato happened to see those papers. And one thing he was thinking about is that. He was thinking, well, maybe one should study this more carefully and try to characterize RNA and find out what's going on. And that probably, he mentioned that in this small letter as an example of what I might consider doing. It turned out those two papers are fraud. Okay? So it was wrong. And some of you may know who were the professor at Harvard. But in any case, it's already quite well known, young professor. And both of them are wrong, fraud, made up. So he was completely wrong when he suggested something very specific to me. But he was right in suggesting, big picture, general direction. And I think this is a very important thing mentors should keep in mind. Mentor should not be just a coach. Mentor should be some vision-giving person. This is the way I feel. And so there are a lot of other things, but I'll stop here, and then I'll let you ask questions.
So when your professor from Kyoto sent you to US, didn't he expect you to go back?
Yeah. So when my visa is going to expire, I did write to him, of course, but I could not. He could not come up with an attractive position for me. So, you mean, do I have a moral obligation to go back? No. I never feel that way. No, he was not unhappy that I didn't come back. He was not unhappy. It was okay. So, people. Well, another message I have with the young people should go abroad. See the world and interact with the different people, and learn from them. I study memory, brain. Brain is so easily shaped by environment. You only think about with the information you have in the head. You have to expand it by going to a new place, interacting with the new people, and exploring new things. But anyway, so.
Becker's lab, produced many other good people. Were people
Renato Drag, yes.
Herman?
Yeah, it's amazing. His lab.
They left when you got there, right?
His lab is amazing. Okay, so now you help me to tell you a little story again. Renato himself is a Nobel laureate, as you know. And his mentor is a Nobel laureate, who is Salvador Luria, okay? And Salvador Luria, the graduate student who overlapped with Renato at the postdoc period in the Salvador lab, is Jim Watson, okay? And in Renato's lab, so David Baltimore, Howard Teming, Lee Hartwell, and myself. At a different time, okay? Lee Hartwell was when he was a Caltech professor, graduate student. So they are the 4 Nobel laureates from his lab.
And I think I remember Paul Berg also did.
Yes, Paul Berg, when he changed from protein synthesis, your molecular genetics, to eukaryotic system, which led to this recombinant DNA technology. He spent 1 year in the Renato's lab for sabbatical. So he was exposed to a eukaryotic system in the Renato's lab. Yeah. So this is quite amazing. We asked him how you do it, how he does it, and of course he doesn't have answer. He doesn't have any answer. That it just happened, he says. The only thing is he does his own research, his own experiment. We had no idea what he was doing, And once every 6 months or so, he sent out notice, not email, no email then, that he's going to give a talk in the lab. So, and he gives a talk, and we don't understand what he's talking about. But he's so far ahead, like, 60, this is the late 60s. He was already thinking, some growth factor in a serum, which will regulate the growth of cells. And he was working under the assumption there is a receptor for the specific growth factor on the cell surface, and then a signal transduction for changing a transcriptional or replicational state. And he was thinking about that. But this is very ahead for everybody else. So we thought, this is, I don't know, this is doesn't look like a hard science. But later we realized that he was just very ahead in everybody else, I think.
Can you describe the telephone call you got from Sweden? I mean, where you were?
I was in bed. It's early morning, so Boston time is a very early morning. So I was sleeping. But also, call was not from Stockholm. Call was from Tokyo, because the reporter called earlier than Stockholm. Anyway, yes, please.
So, if you have to make suggestion to your student or the postdoc, what would be some exciting area for us
For the future?
5, 10 years or
Brain. Well, you should know that everybody, there's no such a thing as objective vision, right? Okay? So each one has it's based on actually based on the memory they have in their head and what is called the taste. And, I consider, brain has a tremendous future. Okay? And brain research can be done many different ways. It's totally interdisciplinary research, right? You can do biological, you can do, physiologically, you can do behaviors, and a lot of new technology is needed also. More technology has to be invented, particularly if we want to study human brain rather than animals. We need a real revolution in a non-invasive technology to look at what's going on in the human brain. Okay? So this is yet to come. MRI is good now, but it's not good enough for real understanding of human mind. Okay? So engineering is definitely there, and I think that's all I can say. I mean, a lot of people say the same thing, but the more I get involved in brain research, the more I'm convinced. And important thing is you are very aware of that interdisciplinarity of brain research. So one should try to learn, trained, not in one level of analysis, but at least 2, maybe 3 levels, multiple levels, so that you can make a connection between processes, events happening in a different level of complexity, cellular cell interactions and physiology, behavior, and so on. But I also say that since this is a biology department, molecular biology will be powerful, continue to be powerful. There's no doubt about it. You know this, right? Molecular biology is applicable for any area of life science. So it's good to be trained as a good molecular biologist, by the way, when you are young. But then you want to think about exploring new problems.
Can you say something about early influences in your life going into a scientific direction? I mean, did you have family members? Did you have a chemistry set? Did you tinker around in the basement, etc.?
Well, that again, I'm confirming what I already said, okay? Human brain is easily shaped by environment, okay? So the doctor's children often become a doctor, lawyer's children often become lawyer, scientists or engineered children often become scientists or engineers, and so on. So I'm not an exception to that. So in my, bigger family and the, uncles, grandfather, they are mostly in science or engineering, including my father. And my brother, my elder brother, became a physicist and so on. So that's the only world I knew when I was younger, teenagers. And it was very natural for me to go into some kind of science. Why chemistry? That's a difficult question. I guess in high school, my grade in chemistry was good. So that's why I thought I may have some talent in there. but that's not, that was just transient.
Can you say a few words about your transition to neurobiology and how you prepared for it?
How? Transfer? Transition?
Transition to
Yeah,
neurobiology.
transition. Okay, so that's another thing. That question is often asked. And, you are young, but not so young. And I'm talking to younger people. You excuse me for that. I think as a scientist, if you want to become a scientist, I think the most important thing is your, I just say, curiosity or your interest, scientific, interest. So, as you do some research in some areas, of course, there are a lot of interesting things there, and you can never really finish it. The question is it really interesting for you? Okay? And, I think people should always think about every few weeks, am I really doing what I'm doing here, research? Is it really worthwhile doing it compared to something else? And in other words, you should always assess, what is called importance, which is also very subjective judgment. Okay? But that's okay. Everything is subjective in this world. Okay? So you should assess whether you are really looking at seeking something really what is called important for you. Important. Okay? And so in immunology, I worked in so-called immunology. By the way, I never felt I became an immunologist, okay? I always felt I'm a molecular biologist. I just used the molecular biology to study immunological problems. So I did that for, 15 years or so, a little bit more. And, of course, there are still a lot of problems. But I started feeling I want to explore something new. I started to explore something new, okay? And there are more subtle thinking on this. And, in science, as you know, if we want to have a really impact to that field that you are working on, I think you need a few things. One is keen interest. And the second thing is some kind of approaches, expertise, or techniques, or something which you like and you think you are pretty good at, but other people are not using in order to study a certain problems in the field. Okay? So it's almost like what is called interdisciplinary approach. Otherwise, I think it's impossible to be able to contribute significantly to a field as a latecomer. You understand? Because there are thousands of, maybe tens of thousands of bright people who have been, thinking about the problems for many, many years. And then you, all of a sudden, you go in there, and if you use the same approach as they are using, or the same thoughts, applying the same thoughts, I think the chance is very small that you can contribute. So you have to run into something that nobody has done. So when I went to Immunology Institute, nobody was doing the molecular biology of nucleic acids. Nobody in the world interested in immunological problem was doing that. And I happened to be trained in that field in other areas. And all I did is combine the problem with the approach I had. Okay? Fortunately, nobody else was doing it. And I did have a competition later, by the way. Okay? Cesar Milstein, Phil Readers, they also came into that. But in any case, so the same thing in immunology. So I felt immunology is coming to the point where the people who have more medical training, medical as well as PhD training, will probably have a better, more fun to continue to study some of the remaining problems in the immunology, like tolerance and so on, autoimmunity and things like that. And I don't have any medical training, and I didn't want to let how do I say? I didn't feel that I'm the one who should be studying all those things. So I wanted to have something new. So there is a push and a pull, okay? The push is what I just said. Pull is I was interested in the brain. I was interested in mind. And, but I didn't know for several years. I didn't know how to study. In other words, I didn't have this approach, which is unique, almost unique to me or to my lab. But fortunately, toward the end of immunological research, we are using this transgenic gene knockout system to study immune system as a system. And then one day, Arsenio Silva, who is now professor at UCLA, came as a postdoc. When I was talking with him, we realized nobody has applied the transgenic or knockout mouse technology to study the mechanism underlying mouse behavior. Okay? Now, the Drosophila people are already doing that. Benza started several years before that. But no, it was now possible also to do with mouse. But nobody was doing that. Never had anybody is starting doing that. So I thought this may be, that was a break with which I went in. We went in. It took about 7 years to phase out immunology and go up neuroscience. But it was a great change because my students and postdocs, many of them also switched. And we learned together. And some of them know better than me. And like MD, postdoc, they know the anatomy of the brain already. They tell me all these foreign names to me. And I learned from them. And not just anatomy, but many things. So it was okay. I had Howard Hughes, so we had stable funding. But actually, Max Cohen, I was on Howard Hughes on immunology program. So when Max Cohen heard that I'm switching, he I don't know what he did. He wrote me or he called me and he expressed his displeasure for my changing it. Because, hey, Susumu, you are in the immunology program.
I can tell you what he said.
Huh?
I can tell you what he said. Because
Oh, what did he say?
Yeah, because I was director of the cancer center
Yes.
at the time when you were making the switch.
Right.
You wanted a room to do electrophysiology.
Uh-huh.
And if you remember, I gave you one.
Uh-huh.
And Max called me up and said, we're not paying for that room. We hired him to be an immunologist.
Yes.
And we don't want him wasting his time.
Right,
praying.
right.
And I said, well, I don't care whether you pay for it. he's got the rub and he's going to do neurobiology.
Good. I didn't know.
He came around later.
Nice. Yeah, came around. He came around. As soon as the first paper was coming out, we immediately changed. They said, "Oh, it's great." So in that sense, Howard Hughes people are very good, very nice to us. Yeah. Yes, please.
What keeps you motivated? I mean,
I can't hear you.
what keeps you motivated to continue the work that you're doing? Is it just the interest in what you're doing, or I mean, just what keeps you motivated to keep going, to keep pushing forward in science? Because some people might have said that you have attained the peak of scientific achievement. So what keeps you going beyond that point?
Oh, I never thought that I'm at the peak. What do you mean? You still have to live. So you want to do something interesting, right? You want to have a fun in your life as long as you're living. So you have to find something which you get excited about, right? So it's great if you change the field late like I did, because you go back, you become student. So you learn a lot of things very fast. It's not like studying in a class and reading a book, textbooks or things like that. That's too slow if we are changing field. Because tomorrow I have to talk to a very well-known neuroscientist for collaboration. So by tomorrow I have to know the basics of what we are going to talk about. So I just ask around the people, and by years we learn things. I mean, it's probably information is not very accurate this way, but at least you get the sound. That's the way I think science should be done. I mean, the science, it's up to you. I mean, if you're interested, you do it. There's no other choice. Otherwise, you're bored to die. Right. I hope it's useful. I'm not sure. It's okay. I will emphasize molecular biology is important, although future is in neuroscience. Yes. Yeah.
I mean, how long do you think it's going to take to actually model the human brain with robots?
Oh, how long it's going to take? I'm sure you can ask that question to somebody else, but you'll get a better answer. But it's tough, okay? I mean, you're talking about a robot who think like human beings. That I don't think it will happen very soon, okay? I mean, actually, it's quite dangerous to do that, because if a robot doesn't like you, I mean, they could have much more power than you have, in a different order of magnitude of power, so they may attack you. And so we have to. I don't think in that way. I don't think a robot will ever be like a human being, because human being will not allow that, right? So, but question is, can robot, without given software, can this machine proactively, not only respond to the stimulation, but have a thought, planning of the behavior? I don't think people have a dream to do that. So I think it will advance. Technology like that will advance, years to come. But to what extent we can do that, I don't know. I mean, and when it depends on what robot you're talking about, what stage of robot. I mean, robot can now take care of patients, for instance, give the pills in the right time and say something soothing to you for the patient. But that's all software is there. I mean, the human is giving them to do that. So they have to create their own. I mean, if you look at the human brain, the human brain hardware changes in response to environment. Okay, hardware cell network itself changes to adapt to the environment. We incorporate information from environment. It's a totally interactive machine with the environment. So please ask someone in artificial intelligence people at MIT. It's very interesting because people who are really hardcore, thinking machine people versus, people who are think about robotics, which is, but also at the same time neuroscientists. Their view is completely different. Okay, so I remember someone. I tell you one little thing and then I'll finish it. Okay. Terry Sejnowski is a very good, respected neuroscientist at the Salk Institute, who is had a very broad understanding, biology to robotics to computational and everything. I really respect him. So he was telling me the other day that some years ago, when a thinking machine at MIT, people were at the height of productivity. He was invited to give a seminar, and on the way to the lecture room, the host professor warned him that not to criticize their robotics or their supercomputers. So he thought about it, and then he said he started. Just about he was trying to start a lecture, little fly flew here. And he immediately thought, you just saw this fly flying here? This little guy can find out where the food is. He knows how to reach there. He knows how to even store it. He can even reproduce himself. Now, I just saw this huge supercomputer in the basement yesterday in here. They can't even move. They certainly cannot reproduce by themselves. Okay? So this was 10 years ago. But so that's the difference of little simple, relatively simple brain and a huge supercomputer. Okay?